All right, this is for students that may not have been in class on the day that I finished up the enzyme lecture and we did the enzyme lab. So, sorry, I'm trying to get my slideshow to work. So what I did here was went through, we stopped right there. So if you watched my last video or you were in class, we stopped on induced fit, and then what induced fit was, remember, is that the substrate's going to bind to the active site of the enzyme, and then the enzyme's going to kind of wrap itself around it. I kind of switched gears here, and I wanted to just talk about what are the things that can affect an enzyme's activity. And temperature, pH, and chemicals can affect an enzyme's activity. And that should sound kind of familiar to you because when we talked about proteins and protein folding, we actually listed these three things that can cause a protein to denature. Since an enzyme is a protein, these are the three things that cause it to denature and that causes it to lose activity. Because if an enzyme is misfolded, or denatured, the active site shape is going to change and the substrate won't be able to fit. Okay, and so I just want to bring your attention to these two charts right here. Notice on this top one that temperature is on the x-axis, rate of reactions on the y-axis. Each one of these curves represents two different enzymes and two different organisms. And so if you look at the black one here, this is an optimal temperature for a typical human enzyme. And you notice it is at highest rate of reaction at 37 degrees Celsius. That should make sense. The optimal temperature for a typical human enzyme is going to be body temperature, which is 37 degrees Celsius. The red curve right here shows a typical enzyme for a thermophilic heat-tolerant bacteria. So like a bacteria that you would find in a hot spring at Yellowstone Park is going to have an optimal temperature at 77. So at 77 degrees Celsius, that's where the enzymes of a thermophilic bacteria are going to work the fastest. So that's called the optimal temperature. Down here, this chart or this graph is showing you pH. Two different enzymes. The black one is pepsin, which is an enzyme in your stomach that digests proteins. Trypsin is an enzyme in your intestine that digests proteins, and what you'll see is that the rate of reaction for pepsin is highest at a pH of 2, which makes sense because the stomach has a pH of 2. Then over on the red one, you'll see that the rate of reaction for trypsin is the highest around 8, which makes sense because the pH of the intestines is around 8. And so anytime the pH, so let's look at this optimal pH for the stomach enzyme. If that pH goes any lower, the rate of reaction is going to go down because the enzyme is misfolding. If the pH goes up and is a little bit more basic, same thing, the rate of reaction is going to go down because the enzyme is misfolding. And then you'll notice up here, and what you should notice with this graph is that an enzyme will lose its activity faster if you go up on temperature, so if you add heat. It will lose its activity if it goes colder, but it's a little bit slower to lose its activity. And so with the lab, we heated up an enzyme and then we cooled down an enzyme, and we found that when we heated up the enzyme, it lost its reactivity a lot quicker than if we cooled it down. And that's just because heat can break hydrogen bonds. All right, so that is what the lab was over with the liver enzyme catalase. And so if you're needing to make up the lab, we will go over that when you make up your lab. Cofactors, so I'm switching gears. Like that was enzyme losing their activity because of those three environmental factors. Now I want to switch gears and just talk about a bunch of different vocab words to kind of finish out this section on enzymes. A cofactor is a non-protein enzyme helper. So it's a non-protein enzyme helper, so it's not a protein. It can be in the form of minerals like zinc, iron, and copper, or it can be in the form of vitamins. So if a cofactor is organic, carbon-based, it is a vitamin like vitamin A, vitamin B, vitamin C. And what they do is they help the enzyme. So what they're doing is stabilizing. So right here you have a coenzyme, it's really big. It's actually working with the enzyme to create an active site, and then a cofactor right here, which is stabilizing the active site. So all I really want you to know is that a cofactor is a non-protein helper, like minerals or vitamins, that help stabilize the active site of an enzyme. So this is why when you're sick, you'll be told to take zinc or vitamin C. Zinc and vitamin C help stabilize the active site on enzymes that function in the immune system. All right, so that is why you need vitamins and minerals. Now you know. All right, now switch gears again. Let's talk about enzyme inhibition. So what I want you to kind of make note of is that a competitive inhibitor and a non-competitive inhibitor, we are talking strictly about drugs and poisons that inhibit enzymes. And I'm going to give you examples of ones for humans that inhibit human enzymes. Two types of inhibition, what that means, it's going to lower the activity of the enzyme, right? So if you inhibit something, you stop it. A competitive inhibitor is going to bind to the active site of the enzyme and compete with the substrate. A non-competitive inhibitor is going to bind to another part of the enzyme, causing the enzyme to change shape, making the active site still exposed, but non-functional. And so pause if you need to write that down, because I'm going to give you an analogy. All right, so a competitive inhibitor would be like if you had your favorite chair in a classroom, because y'all all do that. We all do that. I do that too. We kind of get attached to a chair, a certain place in a classroom, and every time we go into the classroom, that is where we sit. That is our spot. Everybody else knows that's our spot. Well, let's say you come to school one day and you walk in and somebody else is sitting in that chair. That's kind of like what competitive inhibition would be. If the chair is the enzyme and you are the substrate, the person who's sitting in that chair is blocking you from sitting there, they're the competitive inhibitor. And while this is against school policy to do this, if you gathered enough of your friends to come into the class with you, you could probably intimidate that person to leave that chair so that you could sit there. That's how competitive inhibition works. The substrate and the inhibitor are competing for the active site. So we call it concentration dependent, because if there's enough substrate present, it will outcompete the inhibitor, and vice versa. If there's enough inhibitor present, it will outcompete the substrate. A non-competitive inhibitor binds to another part of the enzyme. So this would be like if you walked into the classroom and no one's sitting in your chair, but your chair is broken and dismantled in pieces on the floor. Your chair is available, but it's not in a form that you can actually sit in. So you just don't use that chair. You just find a different one, probably because you don't know how to fix the chair. I think that's actually a pretty good analogy. So just to kind of show you a picture of it, A is normal binding of a substrate to an enzyme. Perfect. B is showing you the competitive inhibitor in blue and the substrate not being able to bind to the active site because the competitive inhibitor is blocking it. C is showing you non-competitive inhibition. The non-competitive inhibitor kind of goes to the back here, binds to this site, changing the shape of the active site. Well, the active site is exposed, it's available, but the substrate can't bind because it's changed shape. So what type of inhibition is concentration dependent? It's this middle one, competitive inhibition. An example of non-competitive inhibition is cyanide. So if you had cyanide poisoning, what cyanide does, I'll go back to the picture. So this is cyanide, it's a poison. It is going to bind to an enzyme that is responsible for ATP production in the mitochondria. So if it binds to this enzyme that's responsible for ATP production, that enzyme isn't going to be able to bind its substrate, won't be able to make ATP. So then the cells don't have ATP. And I think the first thing that will happen with cyanide poisoning that actually contributes to the demise of the poisoned person is that their heart stops because the heart needs ATP to beat. A competitive inhibitor is like a drug, a statin. It's called a statin drug. You don't have to know that, but I'm just trying to give you a real life example of something you may have even seen, because sometimes I'll have students, they're like, hey, my dad takes that or my grandma takes that. So statin drug, Lipitor is one of the most widely prescribed cholesterol drug, one of the most widely prescribed drugs, period, in the United States. I want to say it's still one of the top 5, but I could be wrong. 10 years ago, it was one of the top 5. I haven't looked at the list in a while, but if you look at the top 100 drugs, Lipitor is definitely up there near the top. Lipitor is a competitive inhibitor, and it's going to bind to an enzyme called HMG-CoA reductase. And what HMG-CoA reductase does is it synthesizes cholesterol. So if someone has high cholesterol and they take Lipitor, Lipitor is going to bind to the enzyme that makes and synthesizes cholesterol. Great, now their cholesterol levels go down. So Lipitor is going to function like a competitive inhibitor. So the precursor to cholesterol cannot bind to HMG-CoA reductase, so cholesterol doesn't get made. All right. Now I'm going to switch gears and talk about activation and inhibition of enzymes, but the difference here is these are internal regulation. This means this is naturally occurring in the body and this is normal. So this isn't a result of a drug or a poison. This is something that's naturally occurring inside the body, and it involves regulatory molecules. And regulatory molecules, they're just like little folded up molecules that are going to either activate the active site of the enzyme, inhibit the active site of the enzyme, or something called cooperativity, and I'll get to that in just a minute. So pause if you need to write. Turning my page just to see how much you actually have to write here. All right, and I'm going to show you, this is called allosteric regulation. All right, so right here, you have a protein and then you have an activator that's going to stabilize the active site. Now that enzyme is ready for a substrate to come in. It's active. Then it oscillates back and forth between this active and then this inhibitory form. So here you have the same enzyme, but now the inhibitor has bound to it, making the active site stay closed. All right. It oscillates between the two forms because there has to be homeostasis and a balance. So the activator is going to activate it when it's needed. The inhibitor is going to inhibit it when it's not needed. The perfect example of this is blood clotting. The body has allosteric activators and inhibitors that regulate thrombin, which is the enzyme involved in blood clotting. Pretty cool. All right, cooperativity is another type of allosteric activation, just a little bit different. So you have an inactive form of an enzyme. It has 4 subunits. We're going to call this enzyme hemoglobin. You might have heard of hemoglobin as the protein that carries oxygen. It is considered an enzyme, has enzyme activity sort of. So hemoglobin is a protein that carries oxygen. So cooperativity would mean the first oxygen binds and it makes the second oxygen bind quicker, which makes the third one bind quicker and the fourth one bind quicker. Each oxygen that binds increases the affinity of this enzyme to another oxygen. And what that ensures is that all the active sites are saturated before, let's say this is hemoglobin, before it leaves the lungs. So it's for efficiency. All right, and then the last thing is called feedback inhibition. So go ahead and pause if you want to write all of that down next to feedback inhibition. Feedback inhibition is just another way of regulating metabolic pathways. What it does is what it involves is the end product of the pathway looping back around and shutting down the pathway. Basically making sure that not too much product gets made. It prevents wasting, it increases efficiency. Here's a picture to go with feedback inhibition. So kind of follow along here. The initial substrate binds to the enzyme. Through a series of reactions, that initial substrate is going to get converted into a product. That product, after enough of it is made, one of them or some of them will loop back around, will allosterically bind to that original enzyme. So this is all the same original enzyme. It's just showing you in different forms. It will allosterically bind to that enzyme, shutting down the active site so no more substrate can bind, making more product. So the end product of a pathway loops around, shuts down the pathway so that no more product can be made because enough has already been made. All right, and so that is it for those notes. Please let me know if you have any questions. I'll talk to you later.